It is a real pleasure and honor to be here, and I thank you for coming this evening. I'd like to tell you a little bit about our research over the last 20 years on social isolation. It's interesting that research on individuals, on brain and behavior, really focused on solitary animals, whether they be rodents or humans. From the perspective of many scientists during the 20th century, the contributions of the social world to the behavior and brain were thought to be best considered later, if at all. Now, the rationale was that social factors were of minimal relevance to the basic development, structure, and processes of the brain and behavior. However, social species, by definition, create emergent structures that extend beyond the individual. These emergent structures evolve because they contribute to individual survival, reproduction, and the care of offspring sufficiently long that they too reproduce. Social neuroscience is concerned with the identification of the neural, hormonal, cellular, and genetic mechanisms that support and underlie these emergent structures. If cognitive neuroscience views the brain as the computer and the mind as the software, social neuroscience views the brain as the computer connected to the internet and the mind as the resulting capacities and resources of this connected collective. Now, in the neurosciences, if you want to understand the importance of a particular factor, like a gene, you compare a normal mouse with a mouse with that particular gene knocked out. If I want to understand the importance of a particular part of the brain, I study perhaps Phineas Gage, whose brain I'm showing you here, or before and after the tamping iron obliterated the orbitofrontal cortex. So we took that approach to ask, of what importance are social connections? Of what importance is it to be in the center, a central member of a group, or to be isolated from that group? Now, we started this about 20 years ago, and a couple years prior to it, a study had been published in Science by a group of epidemiologists from the University of Michigan. And what they documented was that across 5 different prospective studies, being isolated from others was associated with broad-based morbidity and mortality, death and disease from a variety of different causes. Now, that was puzzling to them, and in fact, the effect size was as high as obesity, high blood pressure, and sedentary lifestyles. They suggested that the reason was that those who were isolated didn't have anyone, friends or family, to take care of them, to encourage them to engage in better health behaviors. It was called the social control hypothesis. I had trouble with that hypothesis. First, it didn't account for much of the data in the epidemiologic studies, but more importantly, when you look at other social species, they too die earlier when isolated. Fruit flies live shorter lifespans when isolated from other fruit flies. In mice, and you artificially produce an infarct, that is experimentally produce an infarct in the brain, if those mice are isolated, it decreases their survival level and it increases the damage of that experimentally produced infarct. Isolation delays the positive effects of running on adult neurogenesis in rats. It increases the activation of the sympathetic and general medullary system to acute immobilization and cold stressor in rats. And it produces larger morning rises in cortisol in squirrel monkeys. Now, this is just a small smattering of evidence. In every social species we've looked at, isolation leads to faster morbidity and mortality. I doubt seriously if it's because of the health behaviors friends and family of these animals are encouraging. Now, humans are slightly different than the other animals in that they're a fundamentally meaning-making species. And so we find flexible rather than rigid emergent structures. For instance, the emperor penguin creates huddles in the winter, and only in the winter, in order to stay warm. And they voluntarily move inside and go to the outside so that the group survives. Sardines create fish balls when they're predated. These are rigid social structures. Humans come together on occasions like this and disperse and may never see one another again. It extends beyond time and space. All you need to do is to travel in Japan and find how influential dead ancestors are, to the decision-making and reasoning in everyday life to see evidence of that. Interestingly, it extends to ideological as well as genetic similarity. For instance, Manchester United fans have perhaps no genetic similarity and yet feel and share a bond that's like a brotherhood. In the military, they encourage such brothers in arms because it helps the whole group fight and survive the attacks that they must endure. And what I have in the lower right is in Chicago. This is Obama at one of the occasions in which he was speaking, and I, as a member of that group, can tell you that there was a feeling of homogeneity and brotherhood and sisterhood that was really quite stunning. If you've seen him speak, perhaps you can appreciate that. What it means, however, is that perceived isolation and objective isolation are not synonymous. And I have a smattering of pictures here to kind of illustrate that. A little girl who perhaps is watching a parent leave has others around to take care of them. They're not isolated, but they feel terribly alone at that point in time. The student at a schoolyard who's just not in the right clique is around lots of kids and yet doesn't feel a member. The last one chosen for a team, or perhaps an urbanite who's new to an area and walks among thousands of people but feels completely alone. It's the perceived isolation, not the objective isolation, that we have found to be most important in our population-based studies. Don't get me wrong. If you physically isolate someone, you put them in solitary confinement, that will do grave damage. But when you look at a normal population, and we randomly select subjects from the population so we can generalize back to humanity. When we do that kind of research, we find it's not the objective isolation that we've measured that's actually instrumental, it's the perceived isolation. Now, perceived isolation had already been studied in psychology, and it had another term. That term was loneliness. And when we started studying loneliness, it was thought to be like shyness, poor social skills, introversion, or depression. In fact, Robert Weiss, one of the early pioneers in the study of loneliness, characterized it as a gnawing chronic disease without redeeming features. We have found nothing could be further from the truth. We have a bit of a biological perspective, so let me explain our notion of what loneliness is. Genes that promote behavior which increases the odds of the gene surviving are perpetuated, a very simple evolutionary principle. When offspring have long periods of abject dependency, selfish genes lead to social brains. Here's what I mean by that. Our offspring have the longest period of abject dependency of any species. It is not our ability to reproduce that determines whether our genes make it into the gene pool. Now, for many species, sea turtles, snakes, sardines, it is. Their offspring are ready to survive as soon as they're born. Our offspring have very long periods of abject dependency. If there isn't a caretaker who's willing to share defense and food, that infant will not survive. So if we're totally selfish under conditions of privation tens of thousands of years ago, we may live to procreate many, many times, but if our genes have not tricked our brains into caring and bonding and connecting with others, those offspring are not likely to survive. And those genes, although plentifully spread across the land, will not make it to the gene pool because they will not live long enough for themselves to reproduce. So when you have long periods of abject dependency, it isn't your ability to reproduce that determines what makes it into the gene pool. It's the ability of your offspring to reproduce. And that changes how the brain is being manipulated by the genes. According to that notion, loneliness is an aversive biological signal to promote vigilance. Hunger is an aversive biological signal to protect your body. Without that, you may starve or not know that you need food soon enough to search. Pain, similarly, you may, without pain, roll into a fire as you sleep, only to burn and hurt yourself fatally. With those aversive signals, you take corrective action. Loneliness, I would argue, is the same thing. It's an aversive signal that causes you to attend to something that's fundamental to your and your gene's survival. In the short term, it promotes vigilance against social assaults that threaten your short-term survival. If you have disconnections, it may well be because others around you are less trustworthy or are rejecting you or are not valuing you. that puts you in a state of danger. In the long term, it promotes opportunities for social connection necessary for your survival and the survival of your genes. Now, evolutionary time and contemporary time are really quite different places. And at this point in time, culture values the myth of rugged individualism. We think of loneliness as some kind of mental defect or weakness rather than as an inherent trait like hunger, thirst, and pain. If hunger were so stigmatized, we'd have more anorexics walking around. So I'd like to just quickly move through what are we seeing demographically, socially, that might bear on loneliness in the world. And you'll pardon me, I'm going to use US data because those are the data I know best, but I have looked also at China, and I find similar changes in China, although over a much shorter period of time. This is the percentage of single resident households in the United States, state by state, in 1940. The lighter the color, the fewer the percentage of single resident households. And what you see is most of the United States had about 10 to 12% of those households with single people inside. 30 years later, 1970, you see a bluing, a darkening of America. Now we're 15 to 20% in most of the states. 30 years later, 2000, we're approaching 25%. Now, objective isolation and subjective perceived isolation are not the same thing, but indeed, it is a predictor, an inclination that will increase the loneliness across the country. In a sociological study published a couple years ago by McPherson and colleagues, they looked at the social networks of individuals in America compared to what was extant in 1985. And what they found was that the number of contacts, family and friends, had decreased by about 50% over that 20-year period. Even more stunningly to me, they were asked a question: how many confidants do you have? The modal response, the most frequent response they saw in 1985 was 3. In 2004, the modal response was 0. If I had to give you a single question that captured whether someone felt lonely, whether they have confidants, it's not a bad one. Now, when you look at loneliness, I do not want to suggest that this is an isolated variable that travels by itself. So what I have mapped here are undergraduate, young adults, who are lonely and non-lonely, from the top and bottom quintiles of about 3,000 students that we tested. What you see to the right are other dimensions of their personality that also differ. Lonely individuals tend to report lower social support. They tend to report being shyer, having poor social skills, being a little more hostile or angry, being more anxious, lower self-esteem, higher fear of negative evaluation, and poor mood overall. Which of these is the driver and which of these is the passenger? How do we know loneliness is actually the operative variable? And by the way, all of these, in fact, are statistically different. The reason some look closer is some of these scales are shorter, but I wanted to be able to put all of them on one scale. So how do we know when I say loneliness does X, that it's loneliness and not one of these partners in travel? Well, we have 3 methods that we use. In cross-sectional studies like this, we use statistical controls. We measure these other variables and see the extent to which they're accounting for the data. And then if there's any variance left, we see if loneliness is accounting for it. All right? So it's a conservative approach to see if loneliness is the operative variable. More importantly, we do experimental studies where we randomly assign a group like this to become either lonely or not, and we have various nefarious ways to induce feelings of loneliness or feeling together. And I will tell you about such a study in just a moment. But one of the things we wanted to examine was why might perceived social isolation kill you? And when we want to ask that question, we cannot do experiments and follow humans out long periods of time. That would be unethical, obviously. So we do longitudinal studies with all the statistical controls to look for natural variations in loneliness over time to see if an increase or decrease in loneliness is associated with certain physiological outcomes in disease. And so I'm going to be talking briefly about all 3 kinds of studies. Let me begin with an experiment. We had these data I then flew out to Stanford to work with a friend by the name of David Spiegel, who studies experimental hypnosis. And he took a group, or we took a group of 20 high hypnotizable individuals and induced in them, from our study of these individuals, high and low feelings of loneliness. Now, it's the same individuals. I'm going to show you in this next plot. We then gave them all of these scales. And so you will see the same individuals' responses to these scales when they were feeling lonely and when they were feeling very connected. The red and blue lines here are different people. The red and blue lines here are the same people, and the scale is exactly the same. What you see is manipulating loneliness changes who they are as a person, how they view themselves, how they view other people around them. It's really this study, which we did some time ago, that started to convince me we've got something more important than anyone has realized in the past. One of the things it did was to lead to a particular model I'd like to lay out for you briefly. We start with perceived social isolation, which leads to a motivation to connect and, paradoxically, hypervigilance for social threats. Remember, I said there are these dual purposes. You want to protect yourself in the short term from possible assaults from others, and you want to connect with others for long-term survival. And so you have this hypervigilance for social threats. That hypervigilance that you may be completely unaware of, that your brain is doing this, biases attention, cognition, and memory. It also influences how you act toward others. We don't realize the extent to which we're the architects of our own social reality. If I expect a person to be kind, that person will in fact act in a kinder fashion to me. If I expect a person to be hostile, they will act in a more hostile fashion. This has been studied in schools, where if a teacher is led to believe a student is smart, that student performs better that academic year than if the teacher thought the student was not smart. So we see this as a very powerful process in all kinds of domains, including in this social domain. Now, those behavioral confirmation processes produce more negative displays, expressions, interactions, and more negative affect. If I'm interacting with others, I'm more likely to have repulsion and isolation be the result than attraction and inclusion, because of how I'm presenting myself and interacting and interpreting the actions of others. That, in turn, promotes perceived social isolation. This can be a bit of a positive feedback loop that produces chronic loneliness, particularly when our social groupings are so unstable, temporary, rather than living in small villages for long periods of time, like we did in times past. These dispositions, in turn, activate neurobiological mechanisms that increase hypothalamic pituitary adrenal cortical activity and diminish anabolic physiology like sleep quality. That's, in brief, the theory. Let's look at the data. How do you test if your brain is on high alert for social threats and you're unaware of this? Well, fortunately, we have something called the Stroop test. And the Stroop test basically is, tell me what color each of these letter strings is printed in. Well, this is mindlessly easy. Blue, orange, green, red. Okay? Now, I would like you to do that again. Now, again, what the words are is irrelevant. All I want you to do is to tell me the color of each of the letter strings as I present them. You may see that one's harder. And that's because you did not know it, you did not intend it, but you're reading the words. You're attending to the word, despite the fact you wouldn't know it and you wouldn't intend it. So, we reason, let's use the Stroop test to see if there's an implicit hypervigilance for social threats. We've used negative social words like reject, negative non-social words like vomit, positive social words like love, positive non-social words like delicious. And what we did was to subtract how long it said to say orange versus blue and red versus green in these cases. And obviously, all of these words appeared in different colors. There were lots of different words, so it's not color per se that's being named. But we were looking at whether there was something unique about negative social words, rather than just negative words, that impaired a lonely person's ability to say the color. These are the data. When you look at the negative social minus non-social words, lonely individuals were slower at naming the color. They didn't intend to be reading the words, but the negative social words jumped out, got their attention, and interfered with the color naming task. There was no such difference when we looked at the positive social and non-social words, right? Suggesting, in fact, there is this hypervigilance for social threats of which lonely individuals are unaware. What about interactions? This is a study that went 7 days long. Individuals were a beeper, and they were beeped 7 times randomly across those days. When they were beeped, they sat down, they wrote about who they were with, what they were doing, how they felt. What we found was loneliness was associated with more negative affect and more negative interactions. Remember that part of this loop. The quality of the social interactions predicted the affective state. If I had a negative social interaction, I was in a bad mood between that and the next time I was beeped, and vice versa. And we found negative social interactions trumped positive social interactions. Having a quarrel with someone had a longer lasting effect on you and your behavior than having a very positive interaction with that person. Since lonely individuals have more negative social interactions, you can see what this does cumulatively to their thoughts and their life. How does it relate to, if you will, a full social network, like a city? This is Framingham, Massachusetts, the social network status. The darker the color, the lonelier the individual. Every dot's a person. The darker the color, the lonelier the individual. Well, the first thing you can see is that most of us aren't lonely. Right? In study after study, most of us aren't hungry, we're not thirsty, we're not in pain, we're not lonely. These mechanisms are working. They allow us and teach us how, motivate us to reconnect. What we learned, though, is that loneliness lives on the periphery of these social networks. Well, you might say, duh, where else would it be? But the interesting feature is that if you take someone in the middle, maybe one of these bright yellow dots in the middle, if that person becomes lonely, 2 years later, they will be on the periphery and their friends will be lonelier. And the friends of those friends will be lonelier. Loneliness is contagious. Now, it's contagious for the very reason that I just showed you. If I interact with you in a more negative fashion, I put you in a negative mood. You're not likely to interact with somebody else in a more negative mood. As you grow lonelier and more isolated, you become more vigilant for social assaults and find the evidence right in front of you. And over time, you move to the periphery. Well, if there's this hypervigilance, if it's dangerous in primitive times to fend off wild beasts alone with a stick, it is especially dangerous to lay that stick down to sleep when predators are out at night and you don't have a safe social surround. So we hypothesized that this vigilance would be continued through the night, the vigilance of your brain. You don't intend this, but it would actually disturb your sleep. So we measured sleep, and what we found was that lonely individuals fall asleep as quickly. They don't think they do. They actually fall asleep as quickly, but they show poor sleep efficiency. They have micro-awakenings. They're completely unaware of this, but they wake up and then they go back asleep. Sometimes when you travel and you're in a new hotel, you'll have these micro-awakenings because of new sounds. Unaware of how many times you awaken, you'll be fatigued the next day, and indeed, lonely individuals complain of greater daytime fatigue. We also have done this longitudinally, and what we find is that how isolated you feel on an evening determines the next day's feeling of fatigue. Your daytime fatigue does not predict how lonely you feel. So even longitudinally, within individuals, we see feelings of loneliness producing less efficient sleep. Remember, I said a classic position is loneliness is no different than depression. Well, this is what's called a cross-lagged panel analysis. It's a longitudinal analysis. L1, 2, 3, 4, and 5 is loneliness in years 1, 2, 3, 4, and 5 of a longitudinal study we're conducting at the University of Chicago. We have randomly selected individuals from the Cook County metropolitan area. We bring them into the lab once a year and test them. D1 through 5 is depression. Depressive symptomatology, to be specific. I'm predicting depression in year 2 based on what preceded. I'm also predicting it based on psychological status, any antidepressant medications, and physiological conditions. And I take out age, gender, ethnicity, marital status, and education. Only after taking out all of those factors do I see if loneliness can predict above and beyond what all these other factors have predicted. changes in depressive symptomatology? And what you see here is the answer is yes. Loneliness produces subsequent changes, increases in depressive symptomatology. Now, we've also added psychosocial factors, social support that you feel like you have, objective stressors, perceived stressors, hostility. This is a case in point. So this is perceived stress, above and beyond. So we're using all the other factors that I showed you and perceived stress to predict changes in depression. Does loneliness predict anything? And the answer is yes. Nothing that we've put in takes away the effect of loneliness on subsequent depression. Notice depression's not producing loneliness, it's loneliness that's leading to subsequent depression. Now don't get me wrong, not all depression is caused by loneliness. That is not what I'm saying. What I am saying is that the lonelier you are, the more likely depressive symptomatology will worsen. Let's go to the neuroimaging level. Very similar to the Stroop test, where we used social and non-social stimuli to contrast. In this case, we used social pictures that were unpleasant and equally unpleasant non-social pictures. You subtract the hemodynamic response, what the brain is doing when looking at both kinds of pictures, and then you look at what's going on inside the brain. Now, we had 20 such subjects in this study, and what we then did was, after looking at this contrast of each individual in the brain, we convolved their level of loneliness with what was going on in each voxel of the brain. And so what you see here is how did the brains of lonely individuals differ from the brains of non-lonely individuals when looking at unpleasant social pictures, corrected for the unpleasantness per se. Is that clear? So it's how are they responding to unpleasant social pictures in contrast to just something unpleasant. And what you see, this is the back of the brain back here. This is the visual cortices. Lonely individuals are particularly attentive to negative social pictures. Remember, there's this implicit hypervigilance for social threats. Their brain, the visual cortex, lights up, is very active, more so than the non-lonely individual. These blue areas represent the temporal parietal junction. It's an area anterior to the visual cortex, part of the visual stream, and it creates or performs something of a miracle of the mind. It allows you to control your attention, to go from inside your head and your thoughts and your feelings to consider the thoughts and feelings of someone else. When non-lonely people see someone in distress, they're more likely to adopt the perspective of that other person than a lonely individual. They're showing, non-lonely individuals are showing greater activation of the TPJ region. Now, this is understandable. If you're lonely and you feel threatened, a dangerous circumstance raises the need for self-preservation because no one else is around to help. If you're non-lonely, the connection of minds extends to the connection of concerns and empathy and perspective taking. And we did the same thing with the positive stimuli. Positive social pictures, positive non-social, and we find a very different pattern of neural activity. What you see is a large region of activation in the limbic lobe, centered in the ventral striatum. This is a reward area of the brain. Again, it's the non-lonely individuals who are showing greater activity of this dopamine-rich reward region. They're showing stronger activation, stronger, if you will, joy to a social than a non-social pleasant picture, even though they rate both of these equally pleasant. The non-lonely show this response. The lonely actually show greater ventral striatal to the non-social than social pictures. We have a number of autonomic responses I'll hold for questions. Let me tell you a little bit about neuroendocrine responses. I mentioned before, there's a very powerful stress system called the hypothalamic pituitary adrenal cortical system. Cortisol is a very powerful hormone, steroidal hormone, that breaks down fats in order to produce energy. It also is involved in the control of inflammation. Andrew Steptoe here in London showed that lonely individuals show greater morning rises in cortisol. This is our research. We found the same thing as Andrew found. But we did something Andrew didn't, and that is we looked at what other factors might be accounting for this: depression, stress, hostility, social support. Only loneliness was related to this rise. We also looked longitudinally. We asked, how does the day before isolation predict your morning rise in cortisol the next day versus a morning the day before when you didn't feel isolated? And does that morning rise affect that night's level of isolation, of loneliness? And what you see is that the day before loneliness predicted your next morning rise in cortisol. That morning rise in cortisol was unrelated to how lonely you felt that night. So again, we see the same thing longitudinally as we see cross-sectionally. Stronger evidence that it's causal, not just a correlate. Okay, this is kind of a picture of the full system that we've been talking about. And one of the things I'd like to emphasize is we do not find loneliness simply cranks up all stress systems. Loneliness is not related to the parasympathetic or sympathetic discharges. It's not related to the adrenaline, noradrenaline, sympathetic adrenomedullary system in humans. It's uniquely associated with this HPA system, the adrenocortical hormone and the release of cortisol and ACTH. Cortisol courses through your blood, bathing the cells of the immune system, leukocytes, for instance. They have glucocorticoid steroid receptors on them, and it modulates the function of those cells. Sheldon Cohen, for instance, has found that loneliness is associated with impaired immunity. What we asked was, given these leukocytes are bathed in this cortisol, do they develop glucocorticoid insensitivity? This would be a bad physiological outcome. They're no longer sensitive to the signaling that that cortisol is supposed to produce and all the regulatory effects it might have. And so what we did was to look at gene transcripts inside the cells of these leukocytes of lonely and non-lonely individuals. We tested 3,000 transcripts and found, above chance, lonely individuals upregulated 79 and downregulated 131 such transcripts. The green are the upregulated, the red are the downregulated transcripts. Now, this is a large number of transcripts. There are now bioinformatics tools that allow you to look at these transcripts as if they were letters. And based on the functions these transcripts serve, you look to see what words are being spelled out by these letters. What functions are being served. We used the TELUS bioinformatics tool to do that, and what we found was a surprisingly simple answer to the question. Now, I mentioned that there are receptors on these leukocytes for cortisol. They're called glucocorticoid receptors, and the transcript family that takes the signal from those receptors and carries it to the nucleus of the cell are called glucocorticoid response elements. And it is that family of transcripts that's downregulated in lonely individuals. Their cells are responding less to the cortisol signaling that's arriving at the surface of the cell. Now, I mentioned also that cortisol regulates inflammation because the nucleus of those cells are producing transcripts that produce inflammatory cytokines and inflammation in your body. Well, the transcript family that produces that inflammation are NF-kappa beta transcripts, and that was the upregulated family of transcripts that we found. So we see here glucocorticoid insensitivity in people who are chronically lonely. This doesn't occur overnight. This is something that occurs over years. Okay, loneliness affects gene expression, but is loneliness heritable? I would argue we need variations in loneliness. We need those for whom social disconnection is very painful, for those are the ones who care and bond and take care of one another. Really, if you think about it, that's where our humanity comes from. That is who we fundamentally are as humans. But we also need explorers. We need people who are willing to leave everyone else behind, go out by themselves, and discover new things, and then bring that information back. So there's a reason for natural selection of the whole range. We tested this looking at about 8,400 individuals in the Dutch Twin Register with Dorette Boomsma, who directs that study, and found loneliness to be about 50% heritable and about 50% environmental. Okay, to summarize briefly, we have a genetic substrate and we have clear social and environmental factors contributing to whether someone feels lonely or connected. Physiologically, when we're young, at least young adults, we're highly resilient. Young adults don't need flu vaccines typically because their physiology, their immune system is so effective that they can ward off the flu without many difficult symptoms. When you get to be my age, you need flu shots because the flu can kill you. So with time, we have physiological resilience that diminishes. But this descending line isn't equal for all people or for all of us under all conditions. Disconnected minds are more likely to engage in poor health behaviors. Yes, indeed, we have found that as well. The poorer the health behaviors, the faster that decline. Lonely individuals are more likely to incur life stresses and to have fewer assistance from others. They're less likely to even seek it when they are stressed. So that contributes to this decline because the stresses on them accumulate higher. Even when the stressors are the same, they perceive the stressors to be greater and the uplifts in life to be less. And so that contributes to the rate of decline. The stress response itself, specifically this hypothalamic pituitary adrenal cortical axis, is hyperactivated in lonely individuals, which promotes a faster decline. And as I showed you, we not only have energy-producing physiology, we repair and maintain our own bodies. It's called anabolic physiology, and the quintessential anabolic behavior is sleep. And the sleep of lonely individuals, that repair during the night of toxic days, is impaired, diminished in lonely individuals, and that too contributes to a faster decline. Now, it makes sense that if you're going to study social isolation, you would need a village. And I have a village that's global, and so this is really thanks to all my collaborators in the research, and at the bottom are funding agencies who have supported this over the last 20 years. And with that, I want to thank you as well and entertain questions. I think, Matthew, you're going to lead that.